So recording has started, and I hope you guys have seen my screen. Can anyone confirm it?
Yes, ma'am, the screen is
Yes, ma'am.
visible.
Okay, thank you. So in first, as I talked about, we will discuss this article, which was printed in the journal Biofabrication in the year 2015. And in this paper, they have basically used a stem cell that is called induced pluripotent stem cell of human origin, all right, and use them to make many liver tissues using the biofabrication technique of 3D bioprinting. All right. So whatever terms that I just talked about, I'll explain it to them, and in the subsequent slides, it will be more clear to you. So as always, if there is any doubt, you guys can let me know. Okay. So first, let me talk about like how tissue engineering can be used uh, for applications like animal feed drug testing, means an alternative of in vivo animal models, as well as for making personalized medicines. So for these two application areas, tissue engineering is upcoming field. Like you can apply the principles of tissue engineering for these two applications. So, for making artificial tissues, you have to first choose a cell source. So, you can choose either the primary cells that is taken from the human body directly, adult cells, mature cells, or you can use embryonic cells, okay, taken from the embryo and all, and as well as you can opt for stem cells. Okay, so we have already discussed in detail what are stem cells, what are the properties, what are the types, etc. So here also they have talked about the stem cells. The stem cells by nature, they are pluripotent in nature, means they can give rise to multiple types of cell types. Embryonic stem cells are mainly pluripotent, but there are other stem cells which are unipotent and multipotent. But also there are stem cells which are totipotent, like which can give rise to all types of mature cells. So let's talk about the pluripotent stem cells or the embryonic stem cells. So these stem cells can be used to make micro tissues, like miniature tissues, okay, that is which functions and structure is mimicking to that of the native tissue in our body. They can act as a substitute to animals for drug testing because what is happening is all over the world governments are like restricting the use of animals for this kind of drug and cosmetics testing because many protesters are there who want to save the animals. Like we cannot justify the life of an animal for your experiments. So when these kind of concerns, ethical concerns comes up, then we have to look for an alternative that can be used to test the safety of the drugs before they are coming in the market. So another reason being why we need an alternative is there are many drugs that comes into market after preclinical trials and clinical trials. But these drugs are safe when they are tested on animals. But when they are used by humans, like large number of humans, then they often show toxicity. So because of these reasons, these drugs have to be then withdrawn from the market, which basically means waste of money, like billions of dollars goes into development of a drug, as well as the waste of time. Like one drug takes at least 5 to 10 years before it can come to market. Now, after coming to market, it came out to be toxic to humans, so it has to be withdrawn. So it's a huge waste of resources. So in order to circumvent this, many people, like researchers around the world, they are working to make artificial tissue constructs which can act as a substitute to animal models. And these artificial tissue constructs can be used for drug testing as well as for making personalized medicine. All right. Now this is the one thing. Another thing is why should one opt for these kind of pluripotent stem cells? Okay, for making artificial tissues. Now, if you look at the property of these pluripotent stem cells, one is that they are self-renewable, means they can create clones, multiple clones of themselves. So, in this way, you can like culture them in such a way that all the cells remain stem in nature, the stemness is intact. Okay, and you like use them to create the tissue. Okay, and these cells, when they are large number in nature, they still have their stem characteristics. Then you give some special kind of conditions, like with special kind of media, with unique growth factors, etc., which will trigger the differentiation of these stem cells into your desired adult cell type. Okay, so in this way, what will happen because of this self-renewable capability, you will get a large number of cells, okay? uh, which is beneficial when you compare it with primary cells. That is the already mature cell type, differentiated cell that you take from the human body. These cells, they don't divide that much. Means they have a limit to which they can divide. If you remember the Hayflick limit. But when we see the stem cells, their limit of division is much more higher, right? So you can like maintain, if you can maintain that stemness in vitro condition, in culture conditions, then you can create a large number of tissues, okay? And each of these tissues under specific condition will give rise to tissue with differentiated cell type, and then you can use it for your application. So when we talk about pluripotent stem cells, means the stem cells which can differentiate into multiple types of differentiated cells, mature cells. So mainly two types of stem cells come into our mind. First is the embryonic stem cells. Okay, the stem cells taken from the embryo. Okay, another being the induced pluripotent stem cells. So have you any of you heard this term induced pluripotent stem cells elsewhere? Because I recall I haven't discussed it during our discussion on stem cells. But if any one of you have heard it previously, I would like to know.
Yes,
Okay, great. So basically, these two stem cells are pluripotent in nature, are known to be pluripotent in nature. So let me first describe each of these cells, this embryonic stem cells and induced pluripotent stem cells individually, so that if you don't remember what it is, you will help. It will help you. So, embryonic stem cells, it was first isolated from the blastocyst, early human blastocyst in 1998, and it is still done in that way only. So, in the blastocyst, there are two types of cells, the trophoblast and the inner mass cells. The inner mass cells, they are removed, and they are basically the embryonic stem cells. So, these, any tissue construct which are created from these embryonic stem cells, when they are implanted on a person, on a human patient on the side, then the patient needs to take various kind of immunosuppressive drugs because these cells are foreign to our body. Okay, and in order to prevent the attack of our immune system, the patient has to take the immunosuppressive drugs. Also, there are various kind of ethical issues comes up when they talk about the source of these stem cells. So you have to like clear a lot of ethical issues, or just like talk with regulatory authorities, like discuss with them before you can get the embryonic stem cells. Okay. Whereas in this pluripotent stem cells, how they are created is you take some cells, any type of cells from an adult body. Okay, and they are used. Okay, they are only used. You don't need the embryo or anything. You just need any type of adult cells from the body. So for doing it, the ethical concerns, etc., are much less. many less people will object to that. Also, when you're using the adult cells from a patient, and then you are doing some treatment so that it becomes induced pluripotent stem cells, and then again injecting back to that patient's body, then the risk of getting rejected from the patient's immune system will get reduced. Okay, but there is still some chance because you are genetically modifying that cell. So whatever changes that has come up, it can still trigger the immune system. But the amount of immunosuppressive drugs that is needed to reduce that immune system attack is relatively less. So in this way, the use of induced pluripotent stem cells is beneficial in some cases when we compare with embryonic stem cells. Okay, so let me talk about how induced pluripotent stem cells came up. So in 2006, this guy, Yamanaka, so he is a Nobel laureate, and he was given Nobel Prize for the discovery of this induced pluripotent stem cells. So what he did, he took the adult cells, all right, adult human cells, and used retrovirus to transfer few genes of these four transcription factors. Genes encoding these four transcription factors, they were uh, they were made to express into the adult cells. All right. So what we saw is when these four transcription factors expressed at an optimal level, the characteristics of that adult cell it changed. It changed into an induced pluripotent stem cell type. Means it gained stem cell characteristics like uh, self-renewable, pluripotency, clonality. So these polyphenotypic capacity, etc. So these kind of characteristics it gain. So which is why these four factors, custom factors, are also called Yamanaka factors. And so basically, this is how the cell type that is in this pluripotent cells came up. All right, and it is now very like largely used in various labs all over the world as well as for tissue engineering applications. Now, another new technology that this paper is working on or have described is the technique of 3D bioprinting. So 3D bioprinting is we can say a type of 3D printing. I don't know if any one of you have heard 3D printing. Like if you have guys have seen some videos where they are using 3D pen or a 3D printer machine and making household daily items. So that is called 3D printing, where the material is deposited in a layer by layer manner to create a 3D model. Okay, so it is an that is why it is called an additive manufacturing technique where you add some material. There's another called a manufacturing technique called subtractive manufacturing, where a block of material is there and you use lasers, etc., to carve it, okay, and create a 3D model. So this 3D bioprinting is a type of 3D printing, and the only difference is you are going to use this method to create organs, okay, to create tissues using a particular type of cell, a biomaterial, and in several cases, you can add some bioactive factors. So these three things, are the pillars of a tissue, right? So they are combined by this machine, by this 3D bioprinter machine, and we make something called a bioink, which is loaded into that machine. So that bioink moves in XYZ direction to deposit the material in a layer by layer fashion so that you get a 3D model. All right. So there are various types of modes of 3D bioprinting that is available to us in the market today. So there is a droplet based where the machine ejects drops, tiny drops, and the drops are placed in fixed coordinates in XYZ coordinate system. Okay, and in this way, using the drops, you're making a 3D model. There is also where extrusion paste, where you add line, okay, line by line, or you can say filament. Filament are deposited, okay, in a line by line fashion, and in this way, a 3D model is created. And another modality is light based, where light lasers are used to, and it is projected in such a manner on a particular type of material that using light, the material gets cured. It becomes solidified. And when it becomes solidified in a fixed shape, it gives you the 3D model. So when and all of these 3D biooperating techniques, where the cells are being used, cells are living material, right? So cells experience some kind of stress, okay? Like whether you do drop by drop, or you do light line by line, or you irradiate some light. In all this process, cells that are in the bioink or in your material, they experience some stress. So because of this stress, their viability often reduces. All right. So it is one challenge in this area, where people have to use correct type of material, correct type of printing conditions in order to uh, increase the cell viability as much as possible. Because if your funny 3D model does not have enough number of living cells in it, then when you culture it, then you won't get an artificial tissue with the function. The cells won't function properly, all right? And you won't be able to see the characteristics, other type of characteristics that the tissue should have. So, uh, what people, so let me, I have not shown a picture of 3D bar printer, no? So let me show you that, right? So that will be much more clear. Mm, Let me see if there is a video available. Okay. So, don't worry about the sound, okay? Just see how the machine is working. So this is the print head. Okay, this is the print head where there is some bio ink loaded in it, and the bio ink has the cell and the material and biofactory mixture of it. Okay. And the machine is moving in XYZ direction and depositing the material. See, they have written something. So you can give any instruction to write anything, like it can create anything. It is up to you. And what model you design, you just give to the machine the robotic arm that it will move in XYZ direction, deposit the material, and give you the desired shape. Okay, so you can find many interesting videos like this, which shows the printing process. So I hope you guys have understood what is 3D bioprinting, all right, and what are induced pluripotent stem cells. Any query regarding this?
No, no.
All right. So let's now move, enough for the discussion. Let's now move towards the paper. So let's see what authors have done. So in this paper, the authors have developed their own 3D bioprinter. So it is a total of 15 journal, where at that time not many companies around the world are making 3D bar printers. 3D printing technology was there, but the machine which can print cells, and that too in conditions that minimizes the stress on the cell and also sterile conditions. So these technologies were not that much available. Now, what is happening is whoever wants to explore this area, they can just simply purchase this machine because there are many companies available who make world-class quality 3D bioprinters. So, here the authors have developed their own 3D bioprinter, and their machine deposits the bioink on a drop-by-drop fashion. So, their machine is a droplet-based one. Okay, and actually they have called it as valve based one. Okay, so I'll show you. They are talking about what is valve, but here the operation of the printer, which is depositing the material, is happening on a drop by drop fashion. And they have used this 3D bar printer to print human embryonic stem cells as well as induced pluripotent stem cells. And they want to see whether the bioprinting process has compromised the viability, cell viability, or pluripotency. They have also tested whether the printing process is affecting the differentiation of the cells, of the pluripotent stem cells. So they have developed a culturing cell culturing protocol, which ensures the differentiation of pluripotent stem cells to hepatocytes. And they want to see whether the printing process, 3D bar printing process, is causing any changes in the differentiation or not. And then they have used a biomaterial called alginate to make a bioink containing these cells, hepatocyte-like cells. I'll tell you what is this cell, okay? And created 3D structures. These are basically rings, 3D rings, which are 3D, which is having some thickness to it. And later showed that the 3D structures, they are showing the markers that proves that they have formed liver. So hepatic markers means uh, markers specific to livers. So like albumin secretion, etc. They have shown is happening in this 3D printer structures. So what did the authors do? So first, let me go through the methods that they have followed. First is what cell lines did they use? So they have used, already said, two types of cells, human embryonic stem cells and human induced pluripotent stem cells. And these are the cell line names. All right. RC6 and RC10. This is for the embryonic stem cells. And it is for RCI22 and RCI50. So the authors are from the Institute of um, right. Roslin Celab Limited. So this is the company, all right. So this company is based in UK and they are making this cell line, all right. And the authors have used this cell line RC16 and RC10 these cells from them in this study. And this is the diagram of the 3D bioprinter that they have developed and used in the study. So this is the entire cabinet kind of a thing where you can maintain sterile airflow, okay, and it won't allow any external air to come in and contaminate the area, as well as there are print heads. Okay, we call it print heads, into which we load the bioink. So this printer looks something like this. All right. So you supply. So this is the reservoir. That is one tube basically in which the bioink is there, and these are electromagnets. What electromagnets will do? They will control the opening and closing of this nozzle. When electromagnetic field is there, then the nozzle is controlled by some valve, all right? So that valve will open in presence of electromagnetic field. And when it is removed, that valve will close. So at the time of how fast the valve is opening and closing, the drops are getting dispensed. So constantly some pressure is applied. Okay. So this is some gauge that is used to measure the pressure. So constantly some pressure is applied to the reservoir of bioink. And due to the action of these electromagnets, the valve, okay, here some valve is there that is opening and closing. All right. and as it is opening, you will get a droplet. So that is why they have called this bioprinter valve based. Many bioprinters that are available have this similar kind of technology, and we also called it EMD based, like electromagnet droplet based. Okay, so because electromagnets are used, that controls the opening and closing of the valve, and that valve is connected to a nozzle. The nozzle is nothing but a small opening, small aperture, but the valve opening and closing of the valve is important. That ensures when the droplet will be dispensed and when it will be not. Alright, so this is how the bioprinter looks like for them, and how this is how the droplets are forming in their bioprinter. So they have used. So when you're using this machine, you have to set some parameters. Okay, printing parameters we call them. So you have to select the diameter of the nozzle. So this is the diameter that they use. It is pretty constant for all the experimental conditions. Then the pressure. You have to apply some constant pressure to your violin. Okay, then only it will dispense. So this pressure will depend on the viscosity of your biome. So cells and media, when they were using only cell and media, which is basically like watery like, so they're using less pressure. But when you're using hydrogen like sodium alginate, which is slightly more viscous, you have to use bit more pressure. So in this way, you have to optimize. And also there is another thing that is called pulse time. So this pulse time basically refers to the time for which the valve is open. All right, you're giving the electric field to the electromagnets, which is opening the valve, and it is opening like for the first case only for 8 milliseconds. So in this way, the longer the pulse time, the bigger droplets you will get. Okay, the shorter the pulse time, the droplets will be small. So in this are the 3D bar printer parameters. Then they have also explained how they have maintained aseptic condition or sterile conditions while 3D printing. Okay, and they have also tested whether the samples that were 3D printed they were sterile or not. So they have also tested that, and the viability of the cells. Okay, they were tested using a technique called fluorescence activated cell sorting flow cytometer. Okay, so I think we have discussed the application of this flow cytometer when we were talking about the topic differentiation, where a flow cytometer can be used to selectively analyze a cell population based on what kind of surface marker it is expressing. So here, what they're doing, cell sorting is happening. Means uh, surface marker is there. You're using some antibodies, etc., to identify the surface markers and accordingly a cell type. And once they're identified, they are sorted. Okay, they're sorted into two groups, kind of. So you can sort a group of live cells and dead cells. Okay, or you can sort differentiated cells and stem cells in this way using this technique. And for the experiments, they used constant cell density of 1 million cells. Okay. Then they have also developed a protocol that for differentiating the pluripotent stem cells into cells which are hepatocyte-like. Okay, they are not claiming that these cells are hepatocytes only. Okay, because you need they haven't. I think they haven't done extensive gene expression studies for these cells. They have only done one or two studies, which through which they are claiming that they are hepatocyte-like cells. Okay, so there is a difference in the term. That is why the difference in the term. So this is the protocol. Like they for 3 days they have used this media, and then after 3 days they have shifted to this kind of media. Okay, and then for 9 days another media is used. You can see how labor intensive this differentiation process would be, right? And you need so many reagents and chemicals for it. So you have to develop this kind of protocols yourself. before going forward, right? And they have also, as I told you, they have also 3D printed using hydrogel that is called alginate, sodium alginate. So this hydrogel is very interesting actually. It is taken from an algae. It is a plant-based material, and this material, upon when it comes in contact with, say, calcium chloride solution or barium chloride solution, where divalent ions are there, so it changes its state. Okay, because state from liquid to solid state, because these divalent cations, they allow the polymer chains come in close contact, okay, and form, you can say, links in between the polymer chains. These links are called crosslinks. And because of the formation of these crosslinks, you get a solid structure. So we'll talk about more about the biomaterials topic. That is our next topic actually. We will talk about that. So I hope, so they have used this material, sodium alginate, and used the solution of calcium chloride to cross-link it. So what they have done, but one disadvantage of using sodium alginate is it does not contain any moieties like the RGB moieties or anything to which the cell adhesion receptors, the integrin receptors, can recognize and adhere to. Right? But it is because it is a plant-based material, as I told you. It is not from an animal, right? So because of which this material lacks any kind of surface molecules which the cells can recognize, the integral receptors can recognize and adhere to. So people have modified this material by adding peptides called RGT peptides. And these peptides are present in the cell binding sites of the biomaterial. So they have modified this material first and then used that. And in the concentration of 1.58%, they have mixed with no, not 1 million, what tends to be 7 cells, that is cell density that they have used. And they have used uh, hepatocyte-like cells, which are at day 6. So after day 6, so it will be kind of here when it was undergoing hepatic differentiation. So in between this process, they have used these cells. They have not allowed it to complete till day 17. So day 6 cells were used for 3D bioprinting because they want to see whether the bioprinting process is affecting the differentiation process or not. And after differentiation, after 3D bioprinting, you can continue this differentiation, this process till up to day 17, and then they are claiming that you can get a liver-like artificial tissue. Okay. So this is the experimental part of what they have done. So in brief, let me just again summarize. So they have used two types of cells, embryonic stem cells and induced pluripotent stem cells, and that two cell lines each. And they are using a 3D bioprinter, which they have made it on their own. And this bioprinter works on the principle of droplet work, where they deposit the material drop by drop. And two types of printing will be done here. One is where the cells are only mixed with the media, and another is the cells are mixed with the hydrogen like sodium alginate. So when they are mixing with only media, so there will be water like drops will be deposited. So you cannot create what do you say? You cannot lift the structures. Okay, it will break. The cells will disperse. But in the other case, when you are using this hydrogel and then you are cross-linking it, it will be a solid, right, solid thing. You can lift it, right, and it will not break. So these two types of 3D printing that they have done. And also when they are 3D printing using sodium alginate, they have used cells, okay, they have used cells which are at day 6 of the hepatic differentiation process that is being used. Okay. So, is there any query in understanding any of the things that I've discussed so far? This paper is quite interesting actually, and the 3D bar printing process. I chose this paper only because this paper is using 3D bar printing as well as it is touching upon the use of stem cells for 3D bar printing. It's a very upcoming area actually in India and as well as all over the world. Many people are working on this area, so I thought since it's a new area, you guys should be familiar with it. So, is there any queries? Because if you have queries now, then the result part won't be understandable. Is anyone there in the meeting? Nitya, I cannot hear you actually. Is there any query in understanding so far?
No, ma'am.
No, ma'am.
All right, got it. Thank you for like responding. All right. So now we will discuss on the results that they have obtained in doing whatever I discussed. So the first thing that they reported that their 3D bar printer was able to maintain sterile conditions because they have whatever samples that they have printed using the machine, no microbial growth was seen in them. Okay, and they have observed it for at least 3 weeks, like 21 days. They have observed it, and from that they have concluded. So, what if anyone wants to like bit? I would like to go in more details. So, how they do it? We normally do it with our samples as well. So, what we do is we prepare our material, okay, or we whatever they have 3D printed it, okay, so they have 3D printed their samples and they have done it in sterile conditions. Like they have given UV, okay, germicidal UV they have given. Then 70% ethanol is used to wipe around the machines, the nozzles, etc. The culture plates, etc. They were autoclaved so that no microbial remains should be there. And then the samples they were kept in incubator that is set at 37 degrees Celsius and 5% CO2 and it is seen. Okay, after 2 3 days, we observe on the microscope if any foreign thing is growing in it or not. Ideally, it shouldn't. Okay, so when they are observing it for 21 days, from that they are saying that the bioprinter and the conditions in which they have printed their samples, they were completely sterile. Okay, so at that time in 2015, it's a very good thing actually because they have developed their own machine, right? So it's a good result for them. Now, as I said, the machines they are made in such a way that they don't introduce any contamination. All the systems are inbuilt in it. Okay, so only precautions have to be taken by the user to make sure everything is sterile. Okay? Then they have also tested how the pressure, you apply some pressure right on the bioink so that they dispense out. So how the pressure as well as the nozzles that they are using, its length, is affecting the viability of their induced of their pluripotent stem cells. So they have tested it for embryonic stem cells only. So two types of nozzles were used by them. One is short, that is around 8, 8.9 mm. Another is 24.4 mm, long nozzles. So why they have done this test? They have done it because they want to see uh, if you increase the pressure, the cells will experience more shear stress. All right. So because of this cell wall will burst and they will die. All right. So that is why effect studying the effect of pressure is important. Also, if the nozzle length is longer, okay, then the distance for which the cell has to experience that shear from the wall of the nozzle becomes more. People often test the effect of nozzle diameter. If the aperture is very tiny nozzle diameter is there, then again the wall of the aperture will like wall of the aperture will give more stress to the cell wall, and then again cell lysis can happen. So these all factors are responsible that gives shear stress on the cell and thereby affected cell viability. That is why they have tested it. So this is the graph that they have obtained, and where the percentage of live and dead cells are obtained for each of these conditions. So what we can conclude from this graph is the cells that were printed using short nozzles, they had a higher cell viability than the cells printed with a long nozzle. So it makes sense, right? The short nozzle is there, and the cell shear stress experienced by the cell will be less, and thereby the printed cell will more live cells will be there in your printed samples. Another thing, if you increase pressure, okay, if you increase pressure, then also the cell viability is getting affected. We compare the bar chart lengths for 1.4 bar and then zero point 0.5 bar. So for zero point 0.5 bar pressure, the cells that were printed, they were having higher viability than 1.4 bar. So as you increase the pressure, the cell viability is getting compromised. Also, this is the control sample. Control sample means the drops that were not 3D printed using your machine. Rather, you used your pipette. Okay, you normal pipette, the hand pipette that we use to um, add some drops onto your well plates. Okay, so this is the unprinted, a non-printed sample, and this is your printed sample result for the printed sample. Then, and as I already explained, the percentage of live and dead cells, they have done it using flow cytometer in the FACS module, fluorescent activated cell sorting module. They have used that module to analyze the cell viability percentages. Then now compared, okay, they have done a comparison between these two cell population, embryonic stem cells and induced pluripotent stem cells. Like, is there any effect of the printing process on these two types of cells? Okay. So what they report is the difference is negligible. Like both the cell types, when printed, they are having almost equal amount of viability, equal percentage of viable cells are available. So this is the graph actually. So red bar is for the induced pluripotent stem cells. So for the induced pluripotent stem cells, you will see the non-printed cells and the printed cells, they have almost equal number of live cells in them. Whereas for the embryonic stem cells, the printed sample have a higher cell viability percentage than the non-printed one. So the reason that they give is during the printing process, the bioink the pressure that you apply on the ink, because of that, the cells have become much more homogeneous in your drop. So cells are not clumped up because these stem cells, embryonic stem cells, they have a tendency to clump up. And if the cells are becoming clumped, then the supply of nutrients, etc., will be less, right? So the printing process is making the droplets with uh, homogeneous cells are there, means you can see single cells in it. So because of which the viability is increased for the printing process. This is the flow cytometer curve actually, which is that they have also shown in the paper, where the red sections are for the live cells and the dark black dots are for the dead cells. And this is for uh, RC10, okay, control sample and RC10 printer sample. You can see hardly there isn't many much difference that is observable. That is why negligible difference that they have reported. Okay. Now, to validate whether the printing process had uh, any effect on the pluripotency on the embryonic stem cell as well as in this pluripotent stem cells. So, in the previous result, they have shown that there is no effect of the printing process on the viability of these two types of stem cells. The next result, they have shown whether the pluripotency, that is the ability to differentiate into multiple types of cell lineages, that is intact or not. You can say whether or not these stem cells are still expressing the markers unique to stem cells. Okay, because these markers, if they are expressed, then you can say yes, they are stem cells, and you know stem cells will have the pluripotency. So they have observed for the presence of these two markers of ISSEA4. Okay, so these two markers are known for pluripotency. And they also tested another marker, that is SSEA1. This marker is present only in the differentiated cells, not in stem cells. Now, if you refer to the paper, I have not shown the graph because it is a very big graph. So from that graph, it you let me show you. So I'm talking about this graph. This is a very big graph, a detailed graph, where these are the two panels for the biomarkers responsible for showing the pluripotency. Okay, this is the marker for differentiated cells. So what they have reported is for both the embryonic stem cells, so RC6, this is embryonic stem cell, RC10 also embryonic stem cell, and RCI22 is in this pluripotent stem cells. All these cells, whether they are in control, that is non-printed sample, this is for the non-printed sample, and this is for the printed sample, all of these graphs show the similar trend. Like SSEA is not expressed in the pluripotent stem cells. It is not, this graph is towards this side, left hand side, not towards right hand side. Whereas the for of 3 by 4 and SSE4, you can see this graph is positioned like around the center. Okay, and here it is positioned in the uh, right hand side. So these graphs actually show that there is no difference in the expression of the markers. There is no difference in the pluripotencies of these two types of stem cell population due to 3D bioprinting. Okay. So, can any one of you brainstorm and answer why you think 3D bioprinting process like can affect the pluripotency? So, why this result is reportable? Like, why it is so unique that it is published in a paper? Can any one of you think, use some common sense, maybe like why did they opt to or thought to do this kind of a study? I would give you a clue. Clue is the shear stress that the cells experience during the 3D printing process. Anyone?
Ma'am, the shear stress can modify their structure so that they can be
Okay.
uncompatible. I say.
Okay, so you're talking about morphology, cell morphology. It can impact the cell morphology.
Yes, ma'am.
Okay. All right. So you're
And
you're
specifically,
kind of
is like,
yeah. Ethan, sorry, I didn't get the last part. Can you repeat?
and due to pressure, some enzymatic reaction or some enzymes inside the cell can also interrupt its growth. Due to pressure, they can modify.
Okay. All right. So you're pretty close to the real thing. So what I imagine is, and I actually can happen is when a living cell is under stress, any kind of uh, extreme environmental condition like shear stress or anything, then the pattern of genes that is going to be expressed by the cell in order to cope up with this kind of extreme condition, it changes. Some genes gets upregulated, like the stress genes, and some genes gets downregulated. So due to this difference in the gene expression pattern, the cell function, it can change, its morphology can change. All right. So that is why they want to see whether the printing process that the cells are subjected to, whether they are too much harsh, that can like trigger this kind of change in gene expression. All right. So if the gene expression is changed, then you can think like different kind of markers like differentiated cell markers can start expressing. Okay, so you never know, like you have to understand the molecular level things that is going on into the cell in order to give a clear answer to what is going on inside the cell or under like when it is subjected to this kind of extreme conditions. Okay, so I think they could have done a much more detailed gene expression studies, not just flow cytometer study only. But yeah, but still, it helps them prove the point that their process is not affecting the pluripotencies of the embryonic stem cells as well as in the pluripotent stem cells. But a more detailed study of gene expression, like DNA array studies, etc., or mRNA array studies, could have been much more good actually. Now, in this point, they are going to now print the cells. Sorry, here. The next result is on how they have printed the cells which are at day 6 of the differentiation process of the pluripotent stem cells. So in the day 0, they have started with embryonic stem cells. They have optimized this protocol for embryonic stem cells because they say that this protocol, when used for in this pluripotent stem cells, then they are not getting uh, very good cell quality. Okay, means cells are not getting completely mature to hepatocyte, like and not giving hepatocyte characteristics. So this protocol is done for embryonic stem cells. So at day 6, like when it is midway of undergoing differentiation, those cells are harvested, right? Those cells are harvested, mixed with cell culture media, appropriate cell culture media, and loaded into the machine and then printed as blocks into well plates. Okay. After printing, then they resume with the media conditions like here. Okay, till day 17, and then saw whether the differentiation has happened properly or not at day 17. Okay, so in order to check the differentiation is completed, they have checked the presence of some hepatic markers like albumin. Usually, liver cells secrete albumin, then HNF 4 alpha, then ZO. These are some markers known to be expressed by the hepatocytes. So their presence they have tested using technique called immunofluorescence, where you use special antibodies that tag these markers, and these antibodies are themselves conjugated with some fluorescent dye that can be used to uh, see under a confocal fluorescence microscope. Okay, and then you can see whether these markers are there or not because the light. So this is the type of pictures that you often get using the immunofluorescence technique. So A, B, these two panels are for the embryonic stem cells, human embryonic stem cells. Okay, panel A is for the control, panel B is for the printed cells. And you can see both the control, that is the non-printed samples and the printed samples, all all uh, so both types of samples are expressing all three types of hepatic markers. You can see the dot and how this green. So all panels are green because the conjugated fluorescent dye gives emission at around green wavelength. Okay, that is 488 nanometers. That is why you can see green kind of signal coming up in the image. So ZO is used for marking the morphology, right? Albumin is secreted in the cytoplasm, that is when the cell cytoplasm is light up. And HNF alpha is a small amount of protein that is secreted by the cell, so it is coming up as dots. So, sorry, I was wrong previously that they have used only embryonic stem cells because from this figure I can see that they have also worked with induced pluripotent stem cells. Like they have subjected these cells to the differentiation protocol and at day 6 printed them. So these are the panels. C is the panel for the control non-printed sample. D is the panel for the printed sample. And also, they also express all the hepatic markers successfully. So this result helps them to conclude that the 3D bioprinting process, when used to print cells that were in between their differentiation process, after printing, there is no effect on the differentiation. Okay? Cells have continued to differentiate and express the hepatic markers. Okay, so the stress that the cells are experiencing are not causing any change in gene expression, which will cause change in the differentiation part. Okay, the genes that are supposed to express by the cells in order to become differentiated, those their expression is still ongoing inside the cell. The printing process or the stress generated due to printing process is not causing any effect on it. Now, another result is for the 3D bioprinting of these J6 cells in sodium alginate hydrogel. So this hydrogel is basically transparent. When you see it, it is completely transparent. So under light, you can hardly visualize it. So because of which they have mixed this hydrogel with some kind of blue colored dye so that pictures become much more clear. So it is not blue in nature. It is transparent completely. So they have used 1.58% sodium alginate, and the sodium alginate is modified with RGD peptides so that cells can adhere to it and so functions. And they have made this ring kind of structures. So they have deposited droplet in a ring structure, and this hydrogel, when in liquid state, it can spread. It can spread because of which the spaces between the droplets it will become continuous, and you will get a continuous ring structure. And after printing, they have examined uh, how the cells are present, okay, using an assay called live and dead assay, where you use a dye to stain the live cells and another dye to stain the red cells. And under confocal fluorescence microscopy, you can see the live cells colored in green and the red cells colored in red. So, and from that image, you can see how many viable cells are there, okay, live cells are there, and and they have calculated the percentage of viability from those images to be 55%. Means there is a significant loss, right? Significant loss in cell viability that has happened when you are 3D printing it. Okay, because they have done this experiment only 1 hour after printing. So 55% is a huge loss. Accepted percentage of cell viability is around 80 to 90%. That is how you can say that the construct is healthy. Okay, it is healthy. There is more and more number of live cells. But often people can recover this cell viability percentage, means you have to give proper culture conditions. And when you do this kind of live and dead assay, say after 2 3 days, then the cell viability should increase because the live cells, they will proliferate and make up for the lost cells. And then at day 23 of the differentiation process, so they have cultured it. Okay, they have cultured it up to day 23. So at day 6, say the cells were used, and then up to day 17, no, rather they have cultured it till day 23. That is 4 5 days more they have cultured it. And then saw whether the differentiation process has completed or not by checking the presence of some hepatic markers. So this is the graph for the that shows how much albumin is secreted by these kind of structures. So I have skipped to show you one graph. So I talked about the live and dead assay, right? Where 1 hour post printing, they have stained with dyes that stain your live cells with green and red cells with red. So this kind of an image that you can get from a confocal fluorescence microscopy. And from this you can calculate the how many green dots are there, how many red dots are there. Okay. And then they have also used immunofluorescence to check the presence of albumin in the uh, differentiated cells, like after day 23. So these green dots are showing albumin is being secreted inside this. These two graphs I didn't put up in the slide. So I was talking about this slide. So here in this slide, it is a graph basically which has quantified the amount of albumin secreted by the cells that is embedded, that is encapsulated inside the alginate hydrogen in a ring kind of a structure. So there are three samples they have compared. So one is the 2D control sample where you don't use any alginate, only the cell and the media is being printed. Then they have made two types of structures. One ring structure which is having 40 layers, like a tube-like kind of structure like this. So this kind of tube structure where there are layers, so many layers in it. Okay, so one structure had 40 layers, another structure had 20 layers. So more amount of volume basically, and more amount of cells. So the difference being in these three types of sample is in 2D control, the time at which the albumin secretion started, like reached its peak level, it happened at a much earlier stage. Okay, at around like 17, 18 days. Okay, means in between, like around 18 days. At day 18, the aluminum secretion has peaked. Whereas when you look at 40 layered structure, the red one, here it reached around 20 days. Okay, whereas in 20 layer structure, the peak of aluminum secretion is at around 22, 23 days. Okay, so what this helps you understand? It helps you understand that the cells are culturing much more faster, or you can say the differentiation process is faster in 2D than in 3D. It is actually true. When you do 3D cell culture, using organoids or spheroids, the cells uh, experience much more resistance to the supply of nutrients because nutrients and the clearance of waste recycled happens slowly, because of which uh, the cells are not at their exponential growth phase for a very long time. It takes a longer time for them to uh, increase in number and show functionality when in 3D, which is why in 2D the peak in albumin secretion is achieved earlier, right? Whereas when you are making a thick 3D construct, 220 layer tube will be smaller, right? It will be much that not that much thick. Whereas 40 layer structure is more thicker. So when you compare these two structures and how much albumin is secreted in 40 layer structure, the cell number is high. Because of that, the albumin peak albumin secretion happened early, while in 20 layer structure, the cell number is relatively less, right? So the peak is shown at a later stage. But whatever be the case, you are able to see that the 3D printing using hydrogen, I sodium alginate, and the printing condition that you have supplied to it, it is not affecting the differentiation process, right? The cells which were taken in between the differentiation process and then 3D printed and then continued the differentiation process, it successfully gave you uh, artificial tissue structure with hepatocytes in it, right? And you are able to show that the hepatic markers are present, all right? So this result also helps you to minimize the time that is going to take to create a mature artificial tissue using stem cells. So rather than starting only from stem cells and then 3D printing the stem cells and then doing the diffraction process, it will take much more time. Rather, what you can do, you can harvest the cells which have completed, say, 50% of the differentiation process. Okay, you harvest those cells and then you 3D print it, and then for the maturation of the entire tissue, it will take you less amount of time. So initially it was taking you say 17 days. Now it will take you say 4 or 5 days, and after 5 days of from the time of printing, you will get tissue with mature cells of your organ of interest, which can be used for purposes like drug testing or transplantation, etc. So this is this is all this is all from my side, and I hope you guys have found this paper interesting, because this paper talks about the recent technologies that is being used in the area of tissue engineering, right? Like 3D bioprinting, the use of stem cells, right? And the data that they have shown there, it is very very easy to understand. Okay, that is why also that is one more reason why I have chosen this paper for our discussion in this course. So if you have any queries,